In the last video, we talked about passive transport. That is the movement of a substance via diffusion across a membrane, either through the membrane directly or through a channel protein. And those molecules were moving from areas of high concentration to areas of low concentration. But sometimes the cell either wants to or needs to accumulate molecules against a concentration gradient. That means they want to accumulate more molecules inside the cell than diffusion would allow. Because the principle of diffusion works until everything's in equilibrium. That means that it's the same on both sides. So molecules will move from high to low concentration until those concentrations are equal. But sometimes cells want to accumulate things in the cell, or they want to remove things from the cell, such as waste products, toxins, things like that. So if a cell is going to move something against a concentration gradient to accumulate or to remove it from the cell is going to need to expend some energy to do that. So the cells do that using active transport. So active transport means that the cell has to expend some sort of energy. It has to do some work in order to move those molecules into or out of the cell. Now, our book goes into two types of active transport. I'm actually going to lump them all together and just make a big active transport category and just sort of say that in active transport, the cell has to use ATP to transport molecules against their concentration gradient. So if we look at this example here, we are seeing these molecules, these little gray molecules, being pumped out of the cell. Now, if this was diffusion, what would happen? If this was diffusion, these molecules up here in the area of high concentration would want to go in. But the cell doesn't want them in, it wants them out. And it needs to move them from low concentration to high concentration. So it requires energy. Now in our cell, the energy currency is ATP. That is a molecule that stores energy within its bonds, and when those bonds are broken, releases energy that the cell can use to power some sort of job. ATP is far more complex than that, but for now, we'll just go with it. That's all we need to know for this class. So the use of ATP provides energy to power the movement of these molecules from low concentration to high concentration. Now we're actually going to just ignore the secondary active transport for now. I just want you to know that active transport requires energy, and the purpose is to move against the principles of diffusion or against that concentration gradient. All right, so what happens if we have molecules that are too big to move through those proteins? So proteins are pretty large molecules on their own, but they're not the largest. Sometimes there are carbohydrates or sugars or just food particles, or maybe even an entire organism if you're single-celled and you're eating something that you want to bring in to digest. So we have a different mechanism here. So we're moving away from proteins moving things across the plasma membrane, or diffusion moving something across the plasma membrane, and we are moving to a different type of transport. The transport of large particles or a bunch of particles at once is accomplished by endocytosis and exocytosis. Endocytosis, endo meaning inside, cyto meaning cell, so bringing something into the cell or importing something into the cell is endocytosis, or the absorption of large particles into the cell. So this happens by a cell essentially surrounding the particle it wants to bring in, and with the plasma membrane, almost like arms, and engulfing it and pulling it into the cell. Exocytosis is the opposite. This is the export, exo meaning out of the cell. It's the export of large molecules out of the cell. So basically what happens here is the molecules are pushed to the edge and essentially shoved out. So let's look at what this looks like. This is endocytosis. We'll talk about the specifics in just a second, but you can see here, this is a large eukaryotic cell. This is another cell of some sort, looks like a smaller eukaryotic cell, and it is extending around the smaller one and will eventually engulf it, surround it, and then the organelles inside will break that down for its nutrients. So here we have endocytosis. There are actually 3 types of endocytosis, and all 3 work on this same idea of surrounding the particle and bringing it in. So here we have pictured the process of phagocytosis. Now, phagocytosis basically means cell eating. So in the process of phagocytosis, the cell is bringing in sort of solid molecules, or not really molecules, particles. So think of food. Here we've got this picture of the cell. So this is food for this organism that's engulfing it. Here we have this particle, quote unquote particle, that is being engulfed and moved into a pocket-like enclosure into the cell. So the cell membrane sort of wraps around this particle. It produces a vesicle, which is basically a little bubble made up of the plasma membrane and the particle inside. And once in the cell, digestive enzymes from the lysosome, which we'll talk about in the next video, will break this down into its nutrient components. Same thing over here. Once this is fully engulfed, the cell will begin to break it down. We do not have a picture of penocytosis. Penocytosis is cell drinking. So this is when the cell brings in some sort of fluid molecules. Now, one of the things that's important about endocytosis, it is basically non-specific, which just means that the cell is engulfing whatever it happens to engulf at that point in time. That doesn't mean it doesn't have a purpose. This picture here is clearly showing that this cell is attempting to eat the smaller one. But it brings in anything that happens to be in that vesicle. So if there are other little particles surrounding this, they come in too. The third type of endocytosis is called receptor-mediated endocytosis. Now, we talked about those little protein or carbohydrate tags found on cells, and we talked about receptors that are found on cells. So in receptor-mediated endocytosis, this is where the cell is bringing in very specific particles that have the right tags in using the matching receptors. So in this case, the cell is only going to bring in the particles that have the correct tags to recognize their receptors. So this example is actually LDL, which is one of the types of cholesterol found in our body. And our cells need this. Our cells in the liver recognize this and actually have specific receptors to bring in the LDL cholesterol. But just like phagocytosis in the previous example, the cell pinches in or makes an invagination, pinches around it, and encloses in the food particle, or in this case, cholesterol sort of packet into the cell. So cells bring particles or large molecules or food or liquids into the cell via endocytosis, puts it out via exocytosis. So exocytosis is essentially the opposite process of endocytosis. Basically, we have little vesicles filled with whatever molecule is going to be exported. This might be cellular waste. This might be a product destined for somewhere else in the organism. And it is moved from wherever it's produced in the cell to the edge of the cell. The plasma membrane from the vesicle fuses with the plasma membrane from the cell and is essentially, the insides are essentially spit out to be released throughout the body. So this happens to be exactly opposite of phagocytosis. So up here we actually have a picture, a nice example, that is vesicles in the pancreas, which produces hormones like insulin, that are being moved to the edge of the cell to be secreted into the bloodstream. So that's one example of how your cell might use exocytosis.